RNA sequencing or RNA-seq determines the nucleotide sequence of RNA, providing a snapshot of the transcriptome. The results can be qualitative, identifying which transcripts are present and which genes are expressed, or quantitative, providing information about relative gene expression levels, where more transcripts generally indicate higher expression and fewer transcripts indicate lower expression. RNA-seq presents a distinct set of biological, technical, and analytical challenges compared with DNA sequencing. One major challenge stems from the fact that RNA is dynamic and transient, whereas DNA is relatively stable. RNA reflects gene activity in specific cell types at particular times and under defined conditions. Consequently, RNA samples must be collected from carefully selected tissues at precisely the right moment and under well-controlled conditions to accurately capture the biological state of interest. Even then, analyzing gene expression at the cell population level can mask important cell-to-cell differences. Conventional bulk RNA-seq provides an averaged expression profile across many cells, potentially obscuring biologically meaningful variation. In contrast, single-cell approaches resolve gene expression at the level of individual cells, revealing transcriptional heterogeneity. This allows for the identification of rare cell populations, distinct cellular states, and developmental trajectories. Alongside this biological complexity, RNA presents a major practical challenge. It is inherently unstable and highly susceptible to degradation by ubiquitous ribonucleases. As a result, rigorous sample handling, the use of RNase free material, and rapid stabilization or freezing are essential to preserve RNA integrity. A further layer of complexity arises from the composition of the transcriptome. Messenger RNA or mRNA, typically the primary target in expression studies, represents only a small fraction of total RNA, while ribosomal RNA or rRNA accounts for the vast majority. To address this, RNA-seq workflows commonly use either poly-A enrichment, which selects for polyadenylated transcripts, or rRNA depletion, which removes ribosomal RNA while retaining both coding and non-coding RNAs like long non-coding RNAs, small nuclear RNAs, and microRNAs. The choice on whether to use poly-A enrichment or rRNA depletion represents a trade-off between transcript specificity and transcriptome breadth and ultimately depends on the biological question being addressed. Another important challenge arises from the fact that most sequencing platforms and amplification methods operate on DNA rather than RNA, necessitating the conversion of RNA into complementary DNA. During library preparation, RNA is reverse transcribed into cDNA, which is then converted into double-stranded DNA and amplified to generate sufficient material for sequencing. However, this conversion introduces a key limitation. Strand information is often lost. After second-strand synthesis, both DNA strands become chemically equivalent, meaning either strand can potentially be sequenced. As a result, it may no longer be possible to determine which DNA strand corresponds to the original RNA transcript. Stranded or directional RNA-seq protocols overcome this limitation by selectively marking one strand. For example, incorporation of DUTP into the second cDNA strand allows it to be selectively degraded or not amplified, ensuring that only the first strand, reflecting the original RNA orientation, is sequenced. Preserving strand specificity is crucial for accurately resolving overlapping genes, antisense transcripts, and many non-coding RNAs. RNA-seq also presents significant computational challenges during sequence alignment and transcript quantification. Unlike genomic DNA, many RNA transcripts share overlapping sequences due to alternative splicing, gene families, and pseudogenes. As a result, short sequencing reads may map equally well to multiple locations or transcript isoforms, creating mapping ambiguity. This can complicate accurate quantification of gene and isoform expression. Longer or paired end reads, together with splice-aware alignment algorithms, help reduce this ambiguity and improve transcript resolution. Do pause and test your understanding of what we have covered so far by doing an interactive question. The challenges in RNA-seq are reflected in the experimental workflow. In standard bulk RNA-seq, RNA is extracted from tissue, enriched for specific RNA populations or depleted of rRNA, converted into cDNA, and fragmented into smaller pieces if using short-read sequencing before library preparation through end repair, adapter ligation, and amplification. The resulting fragments are then sequenced and aligned to a reference genome or transcriptome for transcript quantification and downstream analysis. Single-cell RNA sequencing differs from bulk RNA-seq primarily in the early stages of the workflow, where individual cells must first be isolated and their RNAs uniquely labeled before library preparation. Each cell's RNA transcripts are assigned a cell-specific barcode, often together with unique molecular identifiers or UMIs during or immediately after reverse transcription. The barcodes allow transcripts to be traced back to their cell of origin, enabling gene expression to be measured at single-cell resolution rather than an averaged population signal. UMIs further help identify original RNA molecules, reducing PCR amplification bias and improving quantitative accuracy. After this cell labeling step, single-cell RNA sequencing follows many of the same fundamental molecular principles as bulk RNA-seq, including reverse transcription of RNA into cDNA, amplification, sequencing library preparation, and downstream sequencing analysis. Following sequencing, cells are computationally grouped based on similarities in their gene expression profiles, typically using dimensionality reduction and clustering approaches. These clusters are then annotated using known marker genes or reference datasets, enabling the identification of cell types, cellular states, developmental trajectories, and transcript isoforms arising from alternative splicing. So that's the workflow for RNA-seq and single-cell RNA-seq. Do pause and try the interactive question on the second part of the video. And if you have found this video useful, do like and subscribe to support the channel. Thank you.